I joined IISER in 2016 as an IPhD student, and I defended my PhD in 2023. And ever since, I've been working in Chennai at a firm called Vantage Research. And my PhD was in condensed matter physics. So, as an IPhD, I joined the physics department. I did my master's coursework there and went on to do a PhD with Dr. Apratim Chatterjee of the physics department. And the broad area of research was soft condensed matter physics. I finished my PhD, defended my thesis in May of 2023. And ever since then, I've been working in a company called Vantage Research here in Chennai. So at Vantage, what the company does, we create mathematical models for drug action, basically. So we develop models for various disease areas such as cancer, diabetes, autoimmune, and so on. And the idea is to best represent what the drug does to the body through a set of differential equations. These are essentially coupled differential equations. And the aim is to help our clients who may be manufacturers of these drugs to make decisions such as what the optimal starting dose in clinical trials should be, what is a good efficacious dose, how to reduce the toxicity, and what are the various trade-offs, and what parameters one can optimize to essentially get the maximum possible efficacy and the lowest possible toxicity. So these are the sort of decisions we help make our clients make. So we have a team of modelers. The background of the modelers are essentially quantitative, so physics, math, engineering. But and we also had a set of biologists who help us in constructing the model. So we also have a team of biologists as well. So I joined IISER Pune right after my bachelor's. I first joined a quantum computing group for the first 2 years. So the program was designed in a way that one has to already select a supervisor right after joining. So I selected a quantum computing group, and after 2 years, I switched to a soft condensed matter group. So in my PhD, what I did was I looked at the organization and properties of soft condensed matter materials. By that, I mean materials which are roughly of the size of tens of nanometers to a few microns. So it is in that length scale that we were looking at. Now, this is a large enough length scale for classical mechanics to be applicable, but this is also a small enough length scale for thermal fluctuations to come into play and techniques of statistical physics to come into play. So that was broadly the length scale I was looking at. And I was interested in how polymers organize, how colloidal particles organize, and in various confining geometries. So that was the broader arc of my research during my PhD. And right now, after having moved to the industry, it's slightly different. What I do is we allow not just me, but we have a team where we construct differential equations, and solving these differential equations sort of help us make decisions. Yeah, so not exactly related to my PhD, but this is a different area. This area is called quantitative systems pharmacology, and it is also in practice under other names. For instance, it's also called mathematical biology and so on. Broadly, I looked at two classes of problems in my PhD. I mean, those are two separate problems. So the first was that suppose there is a polymer chain, and we expect that the polymer to have conformations which are essentially random. By random, I mean each conformation of the polymer chain in space can be mapped to a random walk in 3D. So the monomers are connected to each other. They are adjacent monomers are linked to each other, but they are diffusing. So you don't expect a particular organization or a structural morphology to emerge for a polymer conformation. But it has been observed in nature and also in experiments by some of our colleagues that under certain conditions, a polymer may attain a helical conformation. So the question was, how does this happen, and what are the prerequisites? For example, what does a parameter space look like under which this can occur? And we solved that problem, and we said that the polymer has to have some sort of semi flexibility or rigidity. That means it should resist bending to some degree, and apart from that, the monomers of the polymers can also interact via interactions. So, like there could be interactions such as electrostatic interactions, and even that could be enough to give rise to helical conformations, but for a short period of time, transiently, and they could eventually dissolve. So that was one part of my PhD. And in the second part of my PhD, what we were interested in is to look at the problem of bacterial chromosome organization. So I'll give the broad overview of the problem. The broad overview is this: that for complex organisms such as humans, uh, chromosomes separate via this mechanical forces by these self-assembled structures called mitotic spindles. So they essentially act as levers and they separate the chromosomes. But one of the questions that people had asked for a long time is that, so how does something like E. coli accomplish this, given that there are no mitotic spindles there? And a number of theories were proposed. And one of the hypotheses that a physicist Sukjun Jun made was that if I look at E. coli chromosomes, I can consider them simply as ring polymers. They are a large chain-like molecule which with its ends attached to each other. So they have a ring-like topology. And if there are two such ring polymers in confinement, say in a cylinder, so something very similar to an E. coli cell, say. So if there are two such chains in a cylinder, then these two chains will spontaneously move to two different parts of the cylinder, right? And this is because the whole system tries to maximize the number of microstates, that is the number of ways in which the constituent particles are arranged. So it tries to maximize its entropy. So we see this every day in our daily lives as well. For instance, if we are sitting in this room, we know that all the gas molecules are not clustered in one corner. They're spread out in the entire room, because simply if they're spread out, then the number of ways in which they can get arranged is much more than the number of ways in which they could have gotten arranged if they had been in one corner of the room. So that is the argument. So he said that the two chains will move to two different parts of the cylinder simply because the whole system evolves such that the entropy is maximized or the number of microstates are increased. So so he said that this could happen spontaneously because the same statistical physics law, the law of thermodynamics, which governs all even dead objects, let's say, will govern the segregation of these chromosomes. Now, this was he predicted that geometry is crucial. So the cylinder diameter is crucial, and if the cylinder diameter is made wider, or if the confinement is not strong enough, then the entropic forces also decrease. And this was something that was explicitly tested out by people like Case Decker, who experimentally could alter the diameter of the E. coli cell, and he saw that indeed that the segregation efficiency decreased, right? So now the entropic model of Sukjun was accepted to some degree by the 2010s or so. But what other biologists did was that they did another class of experiments where they tagged certain parts of the E. coli chromosome, and they looked at what the spatial distribution of these parts look like. So are they found everywhere within their cell half with equal probability, or do they have a higher probability of being found in some regions as opposed to others? And what they found was indeed that certain segments are preferentially found in certain parts of the chromosome. But this is something that the entropy model alone of Sukjun couldn't have explained, because if these are just two chains which have spatially segregated, then within each cell half, any of the segments of the chain could be found with uniform probability. There is no reason why a segment would show a preferential likelihood to be in one segment of the cell half. So this is where we came in, and what we had suggested is that the E. coli chromosome is a ring polymer, but we also know that what proteins do is they bridge these distant parts along the chromosome, so they could introduce crosslinks along in the chromosome chain, and that could lead to loops in the chromosome. And why are loops significant? We showed that if there are loops, then the loops would also entropically repel each other. So even as the two chains segregate to two different halves of the cylinder, even in each half, the chromosome also has like internal loops now. So these internal loops would also entropically repel each other, and then the segments would be constrained to some parts of the cell half. So naturally, from the data that the biologist generated of where segments are localized, you could back infer a looped structure of the chromosome essentially. So you can say that, to get this sort of localization data of my segments, you should have this sort of a topology of the chromosome, or this should be the loop structure, and this should be the sites where the crosslinks should be essentially. And what we also ended up finding is that once you get this data and you can match to the experiments, you can also look at the contact map. So what is a contact map? So if you draw a map looking at which segments of the chromosome are proximal to which other segments, then you can draw a map, and you can also, and such a thing is also available from experiments, because experimentalists have already done that with living chromosomes, and the experiment is called a HiC map. And what we found is that even with very few crosslinks, say with 4 crosslinks in a 500 monomer polymer chain, we could arrive at a contact map that is quite similar to that of the actual experimental contact map. When I say quite similar, that means that the broad features of that contact map are there. Of course, the finer details are not there. And we know how to obtain those finer details, because that would require incorporation of more constraints, essentially. But our aim was to show that, this is the principle by which one could obtain the localization data of segments as well, and also get to a contact map which is very similar to that obtained from experiments. So it gives a holistic picture of how the E. coli chromosome is organized. So that was one work, and we also thought, and that led to some other consideration, such as that, this principle is agnostic to the fact that the E. coli chromosome is a living thing. So this entropic repulsion by loops could also happen just as well in synthetic polymers. So can we like design different polymer topologies and see how the segments get localized? How do loops affect the forces of segregation quantitatively? Can we work out what sort of loops would be beneficial for the chromosome from the entropic force point of view? Like finally the two chromosomes have to segregate. Do loops help them to achieve higher entropic forces of segregation? So all of these questions we tackled in a separate polymer physics study. So yeah, so our initial question led to answering a question in biology, and then we went on to a more physics-based question, where we thought that this mechanism could indeed be applicable to synthetic polymers as well. So the experience was, it was fun to sort of enter a new area, but at the same time, I think we as a group also found it hard to read biology papers initially. So we had to rely a lot on physicists who have looked at similar problems and the way they were describing the problem to get a sense of what the problem was and how people have been tackling it. But later on, we have reached out to biologists. One of them is in Amsterdam and the other person is in Israel, and they have been kind enough to listen to us and also give a sense of direction and tell us this is what it means, and uh, also explain to us a lot of the terms. So that also helped. But later on, as we were moving more into the area, I think we got more comfortable with the ideas and we could at least read some biology papers, at least in this area, right? But so the comfort level grew over time. The comfort level was very low initially, but since there were other physicists who had looked into that area, so at least we could rely on that to get a sense of what the problem was. I first enrolled in a BSc Physics Honors program back in 2013. This was at Delhi University, Hansraj College, and I didn't know that something like ICERs exist. So about halfway through my second year, a good friend of mine told me that because from third year onwards there are these entrance exams one writes to get into master's programs in IITs, TIFRs, and so on. So for IPHD courses. So obviously, a bunch of us were preparing for that, and one of my friends said, there are these institutes called ICERs, and they're focused on basic sciences and research, and why don't you think about those? So that was the first time I came to know of ICER and ICER Pune essentially. And then in the third year, I wrote my JAM exam and JEST exam, and then my rank was good enough to be within the cutoff for IISER Pune interviews. And then there were two rounds of interview at IISER Pune, and I was selected for the IPHD program. So I joined as an IPHD student. So I was initially there was this coursework for 2 years, right? So the master's coursework essentially, and I enjoyed the courses, and generally speaking, and the coursework was also challenging in the sense that the questions were not very straightforward. It required some lateral thinking, and so that was nice. And yeah, so I think it helped develop my skills, and ICER also had like a bunch of activities going on all the time because there was a thriving vibrant undergraduate community, right? So there is Carva and all the different clubs, and so there were events all throughout the year which sort of kept me engaged. And so there was something apart from academics as well, so that was nice to have. And because IISER Pune is also in the middle of the city, so one could always go out and have a nice life as well, as opposed to many other institutes are sort of cut off from the rest of the city. Yes, I mean, there are a lot of memories, and those would be with friends, traveling, and all those sort of memories. And yeah, I mean, and even like studying with friends, and that was also often like very helpful. So I felt like ICER always had people to talk about anything if you know who to go to. So like that was nice, and there were so many people who are doing so much interesting stuff, so that was always motivating in a way. Apart from every other thing, so I also used to attend the Kaleidoscope movie screenings. Right. And back in the day it used to be in the guest house. Later on it shifted to the lecture hall complex, but the choice of movies were nice, and the way they were screened were nice. And At least back then there was a discussion just before and after the screening about the movie and the technique. So that would have been nice. I don't know if this is still a standard practice now, but that was quite helpful to understand the movie. So I felt like overall IISER Pune had this nice mix where I could talk to a lot of people. Even, apart from academics as well, there were enough number of interesting activities to keep us engaged. The library was particularly nice. You have books of all sort of disciplines, and even magazines on various things, not just science per se, but it was always like very informative and quite interesting. So you could always like walk out to any of these places and have a nice time. Yes. So, I mean, I have recently been made aware of the IISER Pune Alumni Association ever since the convocation this year took place. So, I think it's a great initiative to bring together people, and IISER Pune is already an old enough institution so that the alumni would be, spread everywhere in various sectors. So it would always be very, very helpful to get their perspective, whether they're in academics or industry, or some, or in some other sectors, to get their perspective, to know about these avenues. For instance, when I was a master's student at IISER, or even in the initial days of my PhD, there were so many industry opportunities that I was completely unaware of. I think the Alumni Connect or Alumni Portal can really bridge that gap. It doesn't matter even if the student wants to do academics, but still, I think the student should be well informed as to what opportunities there are. I felt like I often didn't know that there are many such opportunities, but I think if there's an alumni portal, then any young person who's currently in IISER would actually can look people up on the alumni portal and know about the opportunities that exist. Because often, in academics, we feel like this is the only way, and we don't know what opportunities exist. But it would be good if students also have a sense of direction, and they can choose a path based on their inclination as well.